Hello, welcome to my video on apoptosis, where I will be talking about the two different pathways that trigger apoptosis, which are the extrinsic apoptosis pathway, which is triggered by a ligand binding from outside the cell, and the intrinsic apoptosis pathway, which is caused by certain changes inside the cell. In the first half of the video, I'm going to be focusing solely on the extrinsic apoptosis pathway, simply because it is the simpler pathway. So to begin with, in the extrinsic apoptosis pathway, we are going to have our cell membrane, which I am going to represent in black. And in the extrinsic apoptosis pathway, which is also known as the Fas ligand pathway, we are going to have what we call a Fas receptor in the membrane. And that's going to I'm going to represent that as a single transmembrane alpha helix, singular, with a Fas binding site to bind the Fas ligand in the extracellular fluid. So that's your binding site for your Fas ligand, binding site in the extracellular fluid, and in the intracellular fluid, or in the cytoplasm, which I will name. In the cytoplasm, you're going to have a domain, and that domain is going to be called, which I'm going to represent in blue. That domain is going to be called the Fas-associated def domain, and we're going to abbreviate that for the rest of this video to FADD, Fas-associated def domain. And so how is this going to trigger apoptosis? So what's going to happen is you're going to get a Fas ligand. So a Fas ligand I'm going to represent in green. So a Fas ligand is going to come along and bind to this binding site. And that's going to transmit a conformational change through this Fas receptor. So I should label the whole thing as the Fas receptor. So this whole thing is a Fas receptor. And I'm not going to actually draw that line down there because it's going to ruin the drawing later on. But this whole thing is called a Fas receptor, and when our Fas ligand binds to the binding site, it's going to transmit a conformational change to the FAD, or Fas-associated def domain, Fas-associated def domain. And that's going to allow it to associate with another Fas-associated def domain. Now remember, a domain is just part of a protein that can sometimes appear in many proteins. So this Fas-associated def domain is also going to appear in the second protein. And the second protein is going to be called the FAD adapter protein. And the reason why it's called the FAD adapter protein is because it acts like an adapter and connects these two proteins. It connects the receptor to what we're going to call a procaspase. And a procaspase is basically a precursor of a caspase, and the caspase is going to cause apoptosis or break down our cell. So that's what we're going to get to eventually. But this domain, which is the other part of our FAD adapter domain, is going to be called our def effector domain. And once again, we're going to shorten that to DED, dead, which makes a lot of sense. Def effector domain, dead. And this whole thing will be called your FAD adapter domain. Just like how an adapter to a mains plug, maybe you can connect your UK plug to your EU socket. It allows the connection of two different proteins. And we are going to have our def effector domain dimerize with another def effector domain, another dead domain here. And this dead def effector domain will be a def effector domain attached to a pro-caspase. And the two pro-caspases that can be directly activated by your Fas receptor are going to be your pro-caspase 8 and your pro-caspase 10. So I'm going to represent that as pro-caspase 8-10. And I'm going to draw a pro-caspase here. So the def effector domain, so not every pro-caspase will have that, which is in fact only pro-caspases 8 and 10 will have that, which is why only pro-caspases 8 and 10 can be activated by the Fas receptor. So this is your other dead domain. And your dead domain is going to be attached to the pro domain. It's going to be attached to the pro domain of your pro-caspase 8-10. So that can be either pro-caspase 8 or pro-caspase 10. And your pro domain will be attached to a, I'm going to represent it in yellow now. So we haven't used yellow yet. So your pro domain is going to be attached to your large subunit. Large subunits, my mic's in the way, of your pro-caspase, and then attached to a large subunit is going to be your small subunit. Your small subunit of your pro-caspase 9-10. Okay, and this whole thing, this whole complex of your Fas receptor bound to your Fad adapter protein. So Fas receptor here bound to your Fad adapter protein, or Fad. So that's not that's not a domain, that's a protein, and that protein consists of two domains: the def effector domain joined to your Fas associated def domain. This whole complex of the Fas receptor bound to your Fad adapter protein bound to your caspase, pro-caspase 8-10 is going to be called, so this is a formation, what we've just formed is called the def inducing signaling complex. So these three proteins come together to form def inducing signaling complex, and that happens to, just happens we can shorten that to DISC, D-I-S-C. Okay, so what happens when we form our def inducing signaling complex? But once your pro-caspase 8-10 binds to your def effector domain of your Fad adapter, what's going to happen is your def inducing signaling complex can cleave off this pro domain. So it will cleave here, the cleavage site between the pro domain and the large subunit, and between the large subunit and the small subunit. And that's going to happen, that's how every pro-caspase is activated. You get these two cleavage sites, and that's going to separate all three of these domains, essentially, and subunits. So you're going to get your large subunit and your small subunit separated from the pro domain. And these two subunits are what's going to matter. So they are going to form your active caspases. And we will get to that over here. So after forming the def inducing signaling complex, we're going to free a large subunit and small subunit of our pro-caspase 8-10. And they're going to dimerize. So when I say large subunit, I should actually draw it a bit larger, shouldn't I? So I'll draw a larger subunit. I'm going to draw a smaller subunit just to make clear it is indeed a large subunit. Your large subunits and your small subunits are going to dimerize. So in fact, you're going to get two large subunits and two small subunits joining to form a dimer. So that's two. Essentially, you have two pro-caspases without the. You have two caspases without the pro domain joined together, and that's going to form your active. So they're going to dimerize to form your active caspases. They're going to be your caspases 8 or 10. Okay. So here we have our large subunit and small subunit dimerizing to form your active caspases. Now, what can they do when they're active? So what we've done here, we've only activated a small amount of caspases. You can only activate so many because you can only activate using your Faz receptors, and they're going to only activate a small amount of your caspases, and they're only going to activate your caspases 8 and 10. So how are we going to cause a massive response in the entire cell? How are we going to cause apoptosis, where the entire cell is broken down? Well, we're going to amplify the signal. And how are we going to do that? Well, the caspases 8 and 10. So remember when I said that all the procaspases are activated by cleaving at those two sites. But we're going to have some other caspases. So we should have made something clear that procaspases 8 and 10 we can call the initiator. The initiator caspases, because they start this whole signal, they start this extrinsic apoptosis pathway off. But what we need to activate as well are our executioner. So that's rather befitting, executioner caspases, and they're going to be the ones actually carrying out the sentence. So they're going to be the ones affecting or executing the cell. And remember when I said all the procaspases are activated by cleaving in these two sites between the prodomain and the large domain, and between the large subunit and the small subunit? Well, what our active caspase 8-10 can do is actually cleave the sites of other executioner caspases. So what's going to happen is your active caspases will cleave these sites, free up more subunits, large and small subunits. They're going to form complexes. They're going to dimerize and form more active caspases. And then this cycle is going to repeat. So this active caspase can now cleave another inactive procaspase. So what's that going to be called? That's going to be called a positive. That's an example of a positive feedback loop, because we're amplifying the signal, we're getting more and more active caspases. So eventually you will have built up loads and loads of lots of these active caspases, executioner caspases, which can then break down the different proteins within the cell and then cause apoptosis, which, as you clarify, is the breaking down of the cell and cell death. So these caspases are going to break down proteins within the cell, and of course that includes cleaving more caspases to free up more. So they're going to recruit more troops essentially to carry out the task, recruit more workers. Okay, so that is the extrinsic apoptosis pathway. So I forgot to mention that the Faz ligand can sometimes be found on, so I'm going to draw a cell here, can sometimes be found on lymphocytes. So why can they be found on lymphocytes? Well, because lymphocytes need to kill cells that have been infected. And one way of doing that is by triggering this extrinsic apoptosis pathway, or Faz ligand pathway. It's one way that lymphocytes, white blood cells, are going to be able to kill infected cells. Okay, so now that we've finished the extrinsic apoptosis pathway, I'm going to move on to the intrinsic apoptosis pathway. So I'm going to shift our screen over here. So the intrinsic apoptosis pathway is going to be slightly different. First of all, it's slightly more complicated, and second of all, it's not going to be caused by an outside stimulus. Instead, it might be caused by changes inside the cell. So, for example, DNA damage can sometimes cause a cell to kill itself. So, first of all, we're going to look at how the cell can actually, how how else the cell can actually kill itself. And one other way the cell can kill itself is, so I'm going to draw the cell here, and I'm going to use black as cell membrane as always. But this time I'm not going to draw two layers because what I want to represent is the mitochondria. So the mitochondrial membrane here in red. I'm going to zoom in so you can see clearer. So now, obviously the mitochondria won't be that big, but because what I want to represent is in the mitochondria, today I'm going to draw it slightly bigger. So imagine that as a zoomed in mitochondria. So here we have our outer mitochondrial membrane. And within your outer mitochondrial membrane, you're going to have, of course, have your inner mitochondrial membrane. And between those membranes, you're going to have your intermembrane space. So, and within your intermembrane space, you're going to have what we call cytochrome C. And cytochrome C is, of course, involved in respiration, but it's also involved in, so I'm going to draw cytochrome C here. Cytochrome C. It's also involved in the intrinsic apoptosis pathway because cytochrome C is usually only found in the intermembrane space. So if it somehow managed to leave the intermembrane space, this is going to cause apoptosis. So the whole by activating caspases, which we discussed earlier in the extrinsic apoptosis pathway. So cytochrome C leaving is going to be really bad, which is why the outer mitochondrial membrane is going to be impermeable to your cytochrome C. But that means intrinsic apoptosis pathway can utilize this deadliness of cytochrome C to cause the cell to kill itself. So intrinsic apoptosis pathway is all about getting cytochrome C to leave the cell. And how are we going to do that? Well, we're going to have a set of proteins, and they are going to be from the BCL2 family of proteins. BCL2 family. Okay, and they're from the BCL2 family because the first protein discovered was BCL2, which we will talk about slightly later. And they're all related in structure, so they all contain similar domains but in different combinations. They'll also be missing some of them. BCL2 will have all of those domains, which we'll see later. So first, we're not going to talk about BCL2 first. We're going to talk about the BH123 pro-apoptotic proteins. So BH123 is named BH123 because it has BH1, BH2, and BH3 domains. So we're going to draw We're going to draw 3 domains in different colors, and it's going to have BH1, BH2, and BH3 domains. And it's not going to be in the exact order along the protein. In fact, I think it has BH3 on the very left, on one end. So BH3, and it's going to have BH1, and on the other end it's going to have BH2. So it has these 3 domains. And remember, domains are just conserved protein structures. So parts of a protein, or a particular building block, or a module of a protein that's found in many other proteins as well. So your BH domains are going to be found in many other BCL2 family proteins. And before we move on, let's talk about what BH actually stands for. So BH actually stands for BCL2 homology domains. So your BH1, BH2, BH3 are all domains that were first discovered in the BCL2 protein, but they're also in these BH123 pro-apoptotic proteins, which are under the same family. So that's why they're called BCL2 homology proteins. They're all very similar to the domain first found in this one particular BCL2 protein. So your pH3 123 pro-apoptotic protein is going to contain these three domains that were also found in the BCL2 protein. And what these pro-apoptotic proteins will do, and there are two different proteins we'll talk about from the pH3 123 family, and I'm going to represent them in red. So this is your, I shouldn't represent them in red actually. I'm going to represent them in blue, which is your, I'm going to represent your back protein in blue, and I'm going to represent your Bax protein in green. And these two proteins are members of the BH3 123 pro-apoptotic family. And what they're going to do is, well, they're pro-apoptotic, so they're going to trigger apoptosis. And how are they going to do that? Well, BAC is found in your outer mitochondrial membrane, and Bax is found in your cytoplasm. But what's similar about them, what they're both going to try and do is form a pore. So they can aggregate and form this sort of a pore inside. So each one of these is going to be a BAC protein. And if they do manage to aggregate, they will form this channel pore. So they'll form this BH123 channel. And what's going to happen if we can form a channel in our, so this is your intermembrane space, and this is your, down here is going to be your cytoplasm. What's going to happen if you can form a pore connecting the two spaces? Well, what do we have in our intermembrane space? We have your cytochrome C. Cytochrome C is going to move through the pore into the cytoplasm. And what's cytochrome C going to do in your cytoplasm? It's going to cause the intrinsic apoptosis pathway. What's going to allow it to finish off. So you don't want pores, you don't want them aggregating in your outer mitochondrial membrane. And we should note that BACs can also form aggregates in the outer mitochondrial membrane and let cytochrome C through. But I'm not going to draw it again because it would be the same diagram but in green. So how are we going to stop these BH3 123 pro-apoptotic proteins from forming these aggregates, these pores in the outer mitochondrial membrane and letting cytochrome C out? Well, here's when we talk about the BCL2. So it's when we talk about the BCL2 anti-apoptotic proteins, and as you might expect, they're going to be anti-apoptotic. They're going to prevent intrinsic apoptosis pathway from happening. And they're going to do this by binding to BAC and BACs. But before we talk about exactly what they're going to do, before we draw out them binding to BAC and BACs, we're going to talk about the structure. This is going to be your BCL2 protein. We're actually going to give two examples of BCL2 anti-apoptotic proteins. We have BCL2 and we also have BCLX, and they're going to have the same job, but they're going to have an extra domain. So in fact, they actually discovered four BH domains, or domains that appeared in that were common to many other proteins of the same family. And they are, I'm going to use what color, so I guess I'll use black for your BH4 domain. So here's the extra domain that's in the BCL2 anti-apoptotic proteins, but not in the BH3 123 pro-apoptotic proteins. You have your BH4 domain. You have your BH3 domain, and you also have the other domains. So all of these domains are found in the BCL2 anti-apoptotic family of proteins. And you have your BH 1, and then on the end you have your pH 2. And so here, just to emphasize my point, pH 3 is found in both, pH 1 is found in both, pH 2 is found in both, and pH 4 is found in both BCl2 and BClXL. So that's why they are homology domains, because they've been found in many proteins, and they're homologous or similar, or from a common origin to BCl2. So it probably resulted from some sort of gene duplication and then resulted in divergence. Okay, so now let's talk about what the BCl2 anti-apoptotic proteins are going to do. So to prevent your BH3123 pro-apoptotic proteins from forming these aggregates and pores in the outer mitochondrial membrane, our BCl2, which we're going to represent in yellow, and BClXL, which we're going to also represent in yellow but not coloured in, they're going to bind. Actually, we're all going to represent them in coloured in yellow because they're going to have the same job. They're going to bind to your backs and your back, and they're going to sequester them. So this will, of course, be on the membrane. They're going to sequester them so that they can't form pores. Okay, if they're all bound to something else, then they won't be able to form a pore. And that's how apoptosis is usually prevented. So apoptosis normally can't happen because our pro-apoptotic proteins, our BH3123 pro-apoptotic proteins, are being sequestered, prevented from forming aggregates by your anti-apoptotic proteins, your BCl2 anti-apoptotic protein. They're sequestered. Because obviously you don't want apoptosis going on if everything is normal. So what can trigger apoptosis? So we're going to take a step back and jump to another event to see what can actually cause apoptosis before coming back to our BCl2 family proteins. So what we're going to imagine now is we're going to have our DNA double helix. I seem to be very bad at drawing these double helices, and we're going to get lots and lots of lights, UV light, UV radiation. It's going to cause a double strand break, and there are a couple of proteins that can detect this double strand break, so I should draw a line there to signify a double strand break. So double strand breaks tend to be very bad. They can cause mutations like inversions or ring chromosomes. So all sorts of things can happen if the DNA rejoins to the wrong place. Maybe translocations if it rejoins to a different place. So all sorts of bad mutations, changes in your DNA that can lead to all sorts of diseases like cancer can result from double strand breaks. That's not good. You want to fix that. And if you don't manage to fix that, you want to kill yourself to prevent bad mutations from taking place. So we're going to focus on how we can trigger apoptosis, how we can kill the cell in case we don't manage to fix that double strand break. So we'll talk about how we can fix double strand breaks in another video on non-homologous end joining and homologous end joining, which are mechanisms where we can fix the double strand break. But if that fails, we have to trigger apoptosis. So apoptosis, if we fail to fix, we want to trigger apoptosis. And two proteins that can detect that double strand break are going to be ataxia, telangiectasia, so rather weird name, telangiectasia mutated, and ataxia telangiectasia is a disease. So ataxia telangiectasia mutated just means that this protein is mutated in this disease, ataxia telangiectasia, and we're going to shorten that to ATM because I don't want to keep writing that. And there's another protein, they're going to have the same job, so I'm going to draw them the same. is going to be ataxia, telangiectasia, and instead of mutated, it's going to be associated with RAD50. So RAD50 is another protein that's involved in homologous end joining. We're going to call it ATR for short. So these proteins can detect your double-strand break. So to show that, I'm going to draw arrows showing that double-strand break is going to stimulate or activate these two enzymes. They're going to be enzymes. They're going to be serine threonine kinases. And serine threonine kinases are going to add phosphate groups onto serine and threonine, and both of these have OH groups. So serine and threonine both have OH groups on their side chain, which makes it easy to add a phosphate group onto them because they can get a condensation reaction between the two OH groups. So serine threonine kinases are going to add phosphate groups onto the OH groups of serines and threonines, and in the intrinsic apoptosis pathway, to trigger the intrinsic apoptosis pathway, ATM and ATR are going to phosphorylate another two serine threonine kinases. Should probably change colors now to show that it's the next showcase that it's the next stage. And these two serine threonine kinases are going to, well, again have the same role. They're going to be checkpoint kinases. So checkpoint kinases, and they're going to be checkpoint kinase. We're going to abbreviate them to CHK for checkpoint kinase 1 and checkpoint kinase 2. And they again are going to be serine threonine kinases. And I'm not going to go through what they do again, because they're just going to phosphorylate the OH groups on serine and threonine. And checkpoint kinase, that's going to phosphorylate a very important protein, a protein involved in both DNA repair, so that's causing your double-strand breaks to repair and triggering apoptosis that fails. It's also a protein found mutated in a lot of cancers. It's going to be the famous p53 protein. And your p53 ordinarily, so ordinarily you don't need to trigger DNA repair if the cell's doing fine. You don't need to trigger apoptosis. You don't need to kill yourself if the cell's doing fine. So it's going to be bound to MDM2. And MDM2, what it's going to do is it's going to do ubiquinase. So I'm going to put a U group for ubiquitin. And if you learned about ubiquitination, then you know that what ubiquitin does is cause degradation by this mighty and fearsome protease. Okay, so ubiquitin is a marker. It tells this protease to come and degrade the p53. So p53 will go through this protease. And it's going to get cut up into shreds. Okay, it's going to be shredded up and come out the other end in pieces, like so. So p53 degraded. Okay, so we don't want p53 to halt the cell cycle. We don't want p53 to kill the cell or fix the DNA if the cell's doing fine. But if we have our double-strand break, then we want we want to activate it so that it can do its job. So what checkpoint kinase is going to do, I'm going to draw another p53 bound to I'm going to draw it a bit neater and I can do better. So I'm going to draw another p53 and it's going to be bound to your MDM. But except this time, checkpoint kinase is going to phosphorylate your p53 on a serine, or where is it going to phosphorylate? On a serine or threonine residue somewhere on a p53. And that's going to stop MDM2 from binding. it can't bind, it can't ubiquitinate. So P53 is going to survive. So it survives, it escapes degradation by the almighty protease, and it's going to dimerize. And not dimerize, tetramerize, that's a word. It's going to form a tetramer of P53s. So that's going to be 4 P53s joined together. And in its tetramer form, it's tetramerized. As a tetramer, it can act as a transcription factor. And a transcription factor will increase the expression of certain genes, or maybe decrease the expression of other genes, either directly by stopping the formation, transcription of that gene, or maybe the product itself is going to stop the transcription of that gene. But the P53 is, its role is to, of course, I'm going to put in red, is to stop the cell cycle. So if our DNA is damaged, we don't want to divide, we don't want to copy that DNA because that can cause massive mutations, translocations, if this gets rejoined somewhere else during cell division. So we want to stop the cell cycle, and P21 is going to be the protein transcribes. It's another transcription factor that causes cell cycle to stop. Once we stop the cell cycle, we want to first see if we can fix the problem. So we want to activate DNA repair, but this is not the topic of the video, so we're going to assume that fails. If that fails, you want to go for apoptosis. Okay, if it fails, you can't risk having a mutiny. We can't risk having a cell that's going to be mutated. So we'd rather trigger apoptosis. And how is this going to trigger apoptosis? Well, this P53, I should actually have drawn it apoptosis arrow slightly over here. So how is it going to trigger apoptosis? It's going to upregulate. So this P53 transcription factor is going to bind to the section, the promoter of the gene, and encourage the transcription of another family of proteins. So remember, we had another family of proteins in our BCL2 family, another group of proteins, and they're going to be called the BH3 only proteins. And if you remember what BH3 stands for, it's BCL2 homology domain 3. So it's the same one as in red. And of course, that's going to belong to the BCL2 family of proteins. And they're called BH3 only proteins. Why? Because they only contain a BH3 domain. So they don't contain any of the other BH domains. The only domain they share with your BCL2 anti-apoptotic protein is your BH3 only domain. And I'm going to represent our BH3 only proteins in a colour I've not used. For the BCL2 family, we've used blue for back and green for backs. And then we've used yellow for the BCL2 anti-apoptotic proteins, so that leaves red, which we use to represent the BH3 domain. That's perfect. So BH3 only proteins is going to be in red. That was actually coincidental. I didn't actually purposely leave red for the BH3 only proteins. So let me give you some examples of BH3 only proteins first. So examples of BH3 only proteins are going to be BAD, BIM, BID, PUMA, and NOXA. So here are 5 examples of BH3 only proteins. And what BH3 only proteins is going to do is it's going to bind to your proteins from the BCL2 anti-apoptotic protein family. So if it binds and sequesters proteins, anti-apoptotic proteins, then these proteins can't then go and sequester the BH3, 1, 2, 3 pro-apoptotic proteins. So your blue back proteins on your mitochondrial membrane and your green back proteins in the cytoplasm are free. And what are they going to do when they're free? They're going to form pores. They're going to aggregate and form pores on your outer mitochondrial membrane. Okay, so we're going to have our intermembrane space here, and what do we have in our intermembrane space? We have our cytochrome C. Cytochrome C, and that's going to move out into the cytoplasm. And cytochrome C, when in the cytoplasm, can trigger apoptosis. So now we're going to link our intrinsic apoptosis pathway to our extrinsic apoptosis pathway. So your BCL2 family proteins are going to connect. They're going to let. So quick summary. So radiation can damage your DNA and cause double strand breaks. Okay, that's going to, through a series of serine threonine kinases that can help detect and pass on detect the breakage of the DNA and pass it on to P53. Then P53 can activate or cause increased transcription and expression of BH3 only proteins, which will inhibit BCL2 anti-apoptotic proteins, which means that BH3, 1, 2, 3 pro-apoptotic proteins can let cytochrome can form pores, aggregates in the outer mitochondrial membrane to let cytochrome C outside the cell. So that's how we can translate or convert damage to our DNA to activation of these to let cytochrome C out. But we haven't quite linked cytochrome C to apoptosis. So how does cytochrome C trigger apoptosis? Well, cytochrome C is going to somehow activate our caspases. So remember from the extrinsic apoptosis pathway that our caspases are going to cause this positive feedback loop that's going to increase the number of execution of caspases and destroy the cell. So cytochrome C, what that's going to do is actually it's going to bind to yet another protein, and that protein I'm going to draw here. So here's our cytochrome C, which I now shorten to cyt-C. It's going to bind to our apoptotic protease. So apoptotic proteases are proteins, enzymes that break down proteins responsible for apoptosis. And activation factor 1, and it's going to be shortened to APAP1. So it's one of the factors responsible for activation of enzymes to do with apoptosis. I'm going to draw it like this. And attached to your APAP1, so at one end of your APAP1 is going to be a card domain, and card stands for caspase domain. So caspase recruitment domain, sorry, I got that a bit wrong. Caspase recruitment domain, and we're going to call it card domain. And what cytochrome C can do is actually displace. So notice how it's the same color. It can also bind to this binding site on the other end of your APAP1, and it's going to displace your, so now cytochrome C is bound to your end of the APAP1, and that's going to free your card domain. And your card domain, once that's free, you can actually bind to the card domain of another protein, and that protein happens to be caspase 9. So caspase 9 has a card domain so that it can be activated by the intrinsic apoptosis pathway. It's going to be the only caspase that can be activated by the intrinsic apoptosis pathway. So it's going to begin with as pro-caspase 9. So it's going to have a pro-domain in blue, just like our other pro-caspases. So pro-domain, and it's going to have a large subunit. And of course, it's going to have your small subunit. And binding of your pro-caspase 9 to your card domain. So dimerizations of card domains of these two proteins is going to cause activation. And how do we activate a procaspase we're going to cleave? So when the card domain binds, that allows the cleavage of the large subunit from the prodomain and small subunit. And let me clarify, this is procaspase 9. And what's the cleavage going to do? It's going to release the large subunits, the small subunits into, well, free into the cytoplasm. And of course, they can go dimerize to form active caspases. This time it will be active caspase 9s. So slightly different caspase 9s, but it's going to have the same job. So I should not draw the arrow over there. So this is decided as the intrinsic apoptosis pathway, but they're going to kind of converge. They're all going to cause the activation of more executioner caspases in a positive feedback loop. And the executioner caspases are going to destroy your organelles, destroy your proteins, and end up killing the cell. So that's apoptosis. Your intrinsic apoptosis pathway on the right, and your extrinsic apoptosis pathway on the left. I hope you found that video helpful, and I'll see you in the next video.